Okay. Hi, everybody. Well, I was talking to Mike here and saying it's been an exciting day, a bunch of things going on, but the most exciting was we got a report from that there was a wildfire going on right next to one of our bee yards, and had heard from somebody that we had lost about 120 hives. So my son and I just drove over there just an hour ago and got there, and the fire crew who was cleaning up, they had stopped the fire like less than 5 feet from the edge of the bee yard right there. And so the retardant is sprayed all over the hives, but we didn't lose any hives. So we're very, very happy for that today. California is really on the feeling climate change out here. Our vegetation is changing. We have, due to the induced drought, we have whole swaths of forest just dying, just drying up, turning brown. So obviously these trees who may be 30, 40 years old have never seen drought this intense. And when I talk to the people who replant the forests after forest fires, they are being proactive, and they, instead of taking seeds from the trees at that elevation, they go down 500 feet in elevation and gather seeds from that elevation, because they know that by the time those trees grow up, the climate will have changed so much that it's like going down in elevation. So when a fire burns here, we don't get the same system of plants growing back. We get a system from a lower elevation as it warms up here. So big changes out here. Okay. So let's get back to beekeeping right here. And there's plenty of recommendations that you always hear from beekeepers and what you should do, or people want a cookie cutter, a cookie box recipe. And what I suggest is, instead of asking somebody what to do, you learn how to ask the bees by reading the combs yourself. And the problem, Mike, do you have some, yeah, I guess I'll put you there. You have a number of beginners in this group here?
I can't speak to this particular group, but in the club, we definitely have a lot of first year people.
Okay, so I'll try to make this, cover this with information of use to both beginners and experienced beekeepers. So when I Google word beekeeping, you get 32 million results. And I'll tell you right now, by the time you've gone through the first million of them, you're going to realize there's a lot of conflicting advice out there, enough to drive you crazy. The problem is much of that advice is written by people who really don't know what they're talking about. And so a very interesting scientific paper published twice, 1999 and 2009, is by a couple of scientists called Dunning and Kruger, and the Dunning-Kruger curve. And it's called "How Difficulties in Recognizing One's Own Incompetence Lead to Inflated Self-Assessments." And I made the curve with reference to beekeeping, where the self-assessment of one's knowledge or ability on the vertical axis, and your years of serious beekeeping typically. You start with your first hive, give them some sugar syrup, some foundation, man, they grow up, put on a honey crop, and at that point you think, oh my God, I'm the smartest beekeeper in the world. I don't know what everybody else is talking about, why beekeeping is difficult. That is called the peak of inflated self-assessment right there. And then what will happen typically, like on your second year, when varroa catches up with your colony, and you lose all your colonies to the mite or to something else, starvation, whatever, you go down to the trough of disillusionment. This is very, very typical. And we lose a tremendous percentage of beginning beekeepers at this trough of disillusionment. There are those that do get back on the horse, restock their hives, and they get onto the slope of experience and enlightenment, eventually leading to slow attainment of mastery of the craft. Now, when we talk about mastery of beekeeping, or mastery of anything, you're talking about a minimum of 15 years full time before you can possibly master anything. So people, I see all these people sign up, they would sign up for a beginner's class and then also sign up for the master beekeeper class on the same year. That's ridiculous. This slope of enlightenment and mastery, what's interesting, it never plateaus, it never flattens out. I've been keeping bees for well over 50 years now, And my curve is just steeper and steeper. And I'm learning more and more all the time about beekeeping. And that's to me is beautiful. There's a lot of people though, or some people that never get on the slope. They're first year beekeepers for 20 years in a row. They never learn anything. So you want to be careful about getting advice from others on beekeeping. So let me give you the rules of beekeeping from the bees' point of view, rather than from some human's point of view. Rule number one, they need a warm, dry cavity. When the honeybee moved out of the tropics and started to establish in temperate areas, it needed to maintain a tropical environment, and it did it just as humans did by living inside cavities. And we call them caves or houses, and bees adapted for living mainly in cavities inside hollow trees. So practical application for beekeepers, for bee husbandry, is to provide them a warm, dry cavity. They don't care what they are in, whether it's a Langstroth hive or a Wary hive or a top bar hive or any other kind of hive. They just need a warm, dry cavity. Rule number two, they need food. Their two natural foods are nectar and pollen. Well, nectar for the carbohydrates, pollen for the protein, minerals, vitamins, all those. If they are in an area that does not have nectar and pollen, you can either move the hive to a better area, or you can supplementally feed them with sugar syrup or with pollen syrup, which in many areas is not necessary at all unless you have a specific reason for having the colonies expand at a time they would normally not expand. But that doesn't mean you need to feed them sugar or pollen syrup at any time. And rule number three, they need to manage their parasites. All species have parasites, and all you humans have parasites. You all have parasitic mites living in your hair follicles. Most of you do not have scabies caused by those parasitic mites. So you are in a good host-parasite relationship. With the honeybee, they have a novel parasite, the varroa mite, to which they have not yet evolved adaptation. And then another parasite hijacked in here, the deforming virus, which is what is killing most of the colonies these days. So they need to manage their parasites. If they can't do it themselves, then the responsibility of the beekeeper is to step in and help them manage their parasites, mainly the varroa mite. That's the end of the rules. Let's review the rules from the bee's point of view. Warm, dry cavity, food, manage parasites. End of rules. Anybody else who tells you there's other rules for beekeeping that's a reflection of that person rather than anything that the bees need. Here's my recipe for healthy hives. Keep young, vigorous queens. Unlike humans, in which we know that the females improve with age, that's not the case with honeybees. A young queen will always outperform an older queen. You'll have a lot fewer problems with your hives if you keep young queens. Typically, they're going to get superseded by around 1.5 years of age if you have in a short winter environment. So requeening each year helps a lot. Provide good nutrition, either by keeping them in a good place, moving them to a good place, or providing supplemental nutrition if indicated. Control varroa, not when it gets high, but control varroa all year long. Keep your mite levels low for the entire year. And that's a whole other subject. Minimize toxins in the hive. And that means anything that you dump in there, all the recommendations on the internet of all this stuff you should dump in your hive, we don't dump anything into our hives other than some of the natural miticides in our operation. We don't use any synthetic miticides. Also ag chemicals, you want to avoid as much ag chemicals as possible. But the main thing is to be proactive rather than reactive. So there are beekeepers who are always catching up, and there's other beekeepers who are looking ahead and they avoid the problems in the first place. So my suggestions to you is one, honeybees are amazing, fascinating insects, but they're still just animals. And everything about beekeeping is just basic animal husbandry. So it's the same kind of principles as that. Number two, copy consistently successful local beekeepers. Excuse me, I had chemoradiation for a tumor on my tongue caused by the papillomavirus, and my salivary glands have not yet recovered. So I need to stop frequently to hydrate my mouth, or I can't talk. consistently successful. The sign of a consistently successful beekeeper is not how loud they are or who they talk to. It's that their number one problem is they have too many bees every springtime because they need to either sell or get rid of bees because their colonies are increasing so much. Anybody who is buying replacement bees is not a successful beekeeper. So I would suggest choose somebody who is comfortable working bees barehanded at least, has long experience in your area, and whose main problem is having too many bees every spring. And number 3, understand the 4 horsemen of bee apocalypse. And those are chilling. These are a tropical animal adapted for living in cavities. So chilling is always a stress for bees. Poor nutrition, big problem for because their immunocompetence is reduced with poor nutrition. Toxins, natural and man-made. Out here in California, we have 2 plants that are extremely toxic to bees that we need to avoid. And then parasites, mainly the varroa mite and the associated viruses. Then suggestion number 4 is your management decisions, base them upon the biological needs of the colony rather than by a calendar or what somebody else tells you what to do. And those biological decisions you can determine by learning to read the combs, looking at the combs, and let the bees tell you what they need. So we're going to go through the course of a year here. And I love this quote from T.K. Edwards back in 1920. And what he says is, the best bee master is always the one who most exactly obeys the orders from the hive. And that is us. The good beekeeper is the beekeeper who serves their honeybees. Hey, Mike, could I get you to put your picture on so I can see your face? And then if anybody types in any chat questions, just raise your hand like that, and then I can be happy to answer any questions at any time.
Okay, can you see me now?
I can see you now, right?
Okay.
Yes. Okay, so let's understand the business model, the ecological niche of the honeybee. And they're a very efficient competitor for these sporadic flows of nectar and pollen. They have to compete against all the other pollinators that would be doing that. And they have 2 objectives. And if you understand these objectives, you can understand bees. Number 1 is to store enough food to survive dearth periods. That's why they're called honeybees. It's all about storing honey. This is what the bees want to do. They want to store honey. And 2 is once they've stored enough honey, they want to send out drones and swarms of bees to propagate their genetics. And they'll just keep swarming over and over again once the cavity is full. If you understand that these are the 2 major objectives for the honeybee, then you can know what you can do to help them and what you want to do to avoid starvation, or to encourage honey storage and to avoid swarming. Honeybee gets this jump on the competitors, all the solitary bees and the butterflies and stuff, by having this huge standing army of foragers that cooperate through the communication of the forager dance. And these foragers just sit still inside the hive waiting for the scout bees to tell them to move. Otherwise, they don't waste energy. They don't go anywhere at all. They just conserve the energy. That's a big point. And then all the bees, now here's a colony on the in the process of starving right here. They all share their food equally, and the nurse bees pass jelly throughout all the bees in the colony, and the mid-age bees share nectar or honey throughout the colony. So all the bees know the nutritional status of the colony at the same time, and they all starve pretty much equally, trying to save the queen and the youngest bees and the bees about to emerge. Everybody else is expendable. The queen bee, fascinating insect. Everybody focuses on the queen bee. What you got to realize is she's the only normal female in the colony. She's the default. A female that can reproduce is the norm. The fascinating thing about honeybees or other eusocial insects is all these sterile workers. These have all given up their ability to reproduce in order to serve the queen to get her needs met. The influence of the queen in the colony, she's the pheromonal center. So her pheromones keep the colony cohesive. Other than that, she's just the ovary. She's just the egg layer. She doesn't give any rules or instructions to the colony at all. It's the nurse bees that run the show in the colony. So these are bees from about a week to 3 weeks old, and they control the food supply of the colony. They're the only bees in the hive that consume and digest pollen. And then they convert it to jelly, which they pass via trophallaxis to the queen, to the larvae, and to all the other bees in the hive. So this is the currency of nutrition throughout the hive, is this jelly produced by the nurse bees. So the nurse bees run the entire show. So I plotted out colony population here, number of bees in the hive by season, and showing the nectar and pollen flows here. And this is what we get in our area, where we have a springtime one peaking around June, about mid-June, dropping off a little tiny bit of a fall flow. So there's 4 phases that the honeybee colony goes through. It goes through the buildup phase, when pollen and nectar become abundant. When they get about 2/3 of the way built to maximum population, they go into reproduction phase. That is the swarming, and they will send off as many swarms as they can, depending on their strength. And then at the peak of the nectar pollen flow, they go into the food storage phase. This is where honeybees earn their name, and they store honey reserves to make it through the dearth period, which typically goes, depending upon where you live, through fall and winter. So now dates mean nothing here. Down in Mexico, or over in the Bay Area of California, springtime starts in November, when the nectar and pollen start coming in from eucalypts and plants there. They get their main honey flows then in perhaps February, and then wintertime, the dearth period, starts in July, and from July until November is their dearth period. If you live up in Canada, then your dearth period is over the winter, and people get that confused, meaning that thinking that day length has anything to do with this cycle. Day length has nothing to do with the cycle. Date of the year has nothing to do with the cycle. It's all about the availability of nectar and pollen. I've been down in Australia at the winter solstice, the shortest day of the year, and it's this period right here. Maximum bee colonies are building up to full strength and on major honey flows with the bloom of the white box eucalyptus tree at that time. Okay, so the hive dynamics of the hive population are a function of attrition and recruitment. Attrition is the loss of these workers due to the aging. Aging mainly due from foraging. They only live for about 8 to 12 days typically as foragers, and they wear out. They also, some just wearing out, some by weather or predation. So you have this constant attrition of workers. And then recruitment is rearing the replacement workers. And this is what the colony does as a response to almost any kind of stress, is they just rear more replacement workers. And the formula of recruitment minus attrition tells you whether you're going to grow or shrink. So during the build-up period, a colony gets a net gain per day of around 400 to 600 bees, a net increase in the colony population during this period of maximum growth. Now, the queen is laying about 1,500 eggs a day. So if you're gaining 500 bees a day, I'll let you do this math, Mike. What is the math? How many bees are dying of natural mortality every day? You get 500 bee gain, 1,500 bees, eggs being laid by the queen. So every day, how many are dying of natural attrition? What's 1,500 minus 500?
Looks like 1,000.
1,000. 1,000 bees a day are
1,000.
dying of natural attrition during this rapid buildup period. Keep that in mind when you are loath to sacrifice 300 bees to do a mite wash to save the life of your colony. 1,000 bees a day dying of natural attrition at this time. So that eventually the colony comes into equilibrium at about 400 times the queen's egg laying rate. Hey, Mike, do you have any idea if you can turn off the dinger for the people who are entering? Because that's all going on to your recording, I imagine. It's just constant.
Yeah, let me check.
Okay. I haven't heard of, I don't know if anybody's figured out how to do that. Once you get up to be about 40 times the queen's egg laying rate, the colony comes to equilibrium. So if a queen's laying 1,000 eggs a day, attrition and recruitment come to equilibrium about 40,000 bees. That's about a double deep full of bees. If the queen's laying 1,500 eggs a day, then you can have 3 deeps completely full of bees. You get up to 60, maybe 70,000. And then when the queen starts to knock off egg laying later in the season during the dearth period, then the attrition exceeds the recruitment and the population goes down. And the age structure of the colony changes over the course of the year. So this is a graph from data from Lloyd Harris in Manitoba, Canada, showing how you get very, very old bees during the winter. You get bees up to 250 days old. They're almost immortal during the winter. And then come early spring, late winter, they start rearing the new bees. So the red are bees that are 0 to 12 days of age. These are 13 to 24 days of age, 25 to 36 days of age, and very few of them live past 48 days during the summer period. But notice these numbers down here are your average age of the bees in the colony. So your average age here is 128 days, a very geriatric population. Average age through most of the summer is anywhere from 15 to 24 days of age. So the colony may look the same, but you're looking at very different bees. During the spring and summer, you're looking at a bunch of very, very young bees. During the dearth periods, you're looking at more older bees in the hive. And I can break this down, this figure here, of different phases, biological phases. And I have a whole bunch of articles, about 13 articles called Understanding Colony Buildup and Decline on my website. And you go through these various stages. We're going to go through these very quickly with photographs now throughout the season. But let me give one more suggestion. Prior to varroa, the advice I would give the beekeepers is, if you don't know exactly what you're doing in the hive to help the bees, just don't do anything. They'll take care of themselves. Now with varroa, you do need to step in and manage varroa. But other than that, before you take the lid off a hive, have a plan. Know exactly what you're going to be looking for and what you're going to do if you see it. Don't just open a hive and start just screwing around in there. Understand what you're looking for and what you're going to do. Get your plan there.
Yeah.
And
So listening to this, it's really good. I really want to hear it.
What's this white thing?
What white thing?
There you go. You got him? Okay. Experienced beekeeper, you do an entire hive inspection in about 15 seconds. It's very, very quick. The other thing to understand is every club, I suggest, make a chart like this with the approximate dates. Now, here where I live, where we have huge changes in elevation, dates mean almost nothing. I can drive a half a mile from my house and the blackberries start blooming a full month before they do a half an hour in the other direction. We can get the blackberry flow, honey flow at 2 times in the same season by moving those bees a half an hour from a lower elevation to a higher elevation. So the dates aren't so important. What you look at is the phenology, the put in when the different plants are blooming. Then any beekeeper in your area can plan what they're going to do based upon the phenology of the plants. Put in when you have dearth periods, when you have the swarming period. It's typically about 60 days of buildup from winter cluster until colonies are ready to swarm, and another 30, 60 days before the main honey flow. Again, that varies a lot. I know some of you people in Virginia out in the forests, you have very, very early honey flows. So again, all beekeeping is local, but make this chart up for the beginners. Keep some records over a couple of years of when the plants are blooming and overlay colony population and events with that. Very helpful. And then over that, you can overlay what kinds of management you would do. So again, for my area where I live, I show this to my beginners of when you got to get on varroa, when you're trying to prevent swarming, when you're going to have European foul brood inside the colony. You're not going to have it the rest of the time. when you have to do the final cleanups, then you might want to give them some extra supplemental feeding to get ready for the winter. So something like this would be very helpful. If you look on my first year beekeeping at my website, most all of these graphs are on there. And then understand what's happening in the colony on reading the combs, that you can recognize this is aged honey because the airspace below the cappings has dissipated out. And now the capping then touched the honey. As soon as the capping touched the honey, it looks dark. Otherwise, it looks white. So this tells you this honey is aged here. This is fresh pollen, dull looking, dull bright color, and then bee bread, which is fermented stored pollen up here. So this shows that there's a nice reserve here of pollen in the hive. Then you have your open brood, your eggs and your larvae, which are being fed here. And the sealed brood, which is also known as varroa food. This is where varroa is reproducing. Anytime you see a sealed brood, you have varroa mite reproduction in your colonies. Now, I told you the nurse bees run the show. And how do they know what's happening outside the hive? Let me give you a view of what it looks like inside the hive to the nurse bees. That's the view of the nurse bees. You got to realize that honeybees live most of their life in total darkness without seeing anything. And then they suddenly start flying outside into daylight. So the nurse bees need to respond to the cues coming in from the foragers. The incoming pollen, whether there's fresh pollen being put into the middle of the brood nest, whether there's incoming nectar or whether the colony is breaking into the honey reserves, and whether you have larval pheromone here showing there's young larvae being reared. And the interface to pay attention to is this interface between honey and the brood. You can see here's fresh honey, still has the airspace, and this honey is getting older and the capping is reducing down. So this critical interface right here. What you're seeing is the energy reserves, energy stores here, which are placed above and to the sides of the brood nest. Here's your brood down here, which is your energy and protein demand for the hive. And this is your reserve, your larder, where any unconsumed pollen remains. This is not where the pollen is preferentially stored. It's stored right here in the middle of the brood nest. But this means that you have unconsumed pollen, that there's more pollen coming in than the nurses needed to eat at that time. Your energy interface is this single row of cells between any stored bee bread and the stored honey. If these cells here are full of open honey, then that means the bees are eating up into the reserves. If instead these are full of fresh nectar, which shakes out of the comb easily, that means that they're pushing the brood nest downward as they're storing energy reserves. Now, this is your protein interface. This is your suggestion of whether or not you have a surplus of pollen coming in. If this interface disappears, that means they're living hand to mouth, and they don't need to have a reserve. They can thrive and build up without any reserve. But if you don't see a pollen band, that means they don't have any reserve. And you can see here where I've crushed bees in water in order to dilute their gut contents to look for nosema spores, that the nurse bees, that water turns yellow with the pollen that was in their guts, whereas the foragers, it remains clear because the foragers don't consume pollen. Only the nurse bees consume pollen. And then those nurses then share that, convert that pollen into jelly, which is then shared throughout the colony. Okay, let's go through the year, 4 seasons here. Okay, we're going to start first thing in the spring. The first tree pollens that come in my area, it's alder pollen coming in, starts coming in first or second week of January. And this is the cue to the nursery bees that it's springtime. It's time to start rearing brood. And up in Manitoba, Canada, that happens right about here. This dotted line is the amount of brood in the hive. You can see that it declined to almost nothing, and then it starts to build up here when the first tree pollens come in. And this gives a shot of fresh protein. So this is taken in January. This is a fresh alder pollen coming in to my hives, and the bees immediately ramping up a brood rearing here. That pollen coming in, stimulates the nurse bees to produce jelly. And then the pheromones from these very young larvae, here's a second instar larva right here. Yeah, you have a pheromone called E-beta-osmine, and that tells every bee in the hive that the queen is laying eggs and that the nurses are rearing young larvae. So every bee in the hive is aware of the state of buildup of recruitment in the hive. We'll get back to this during swarming cue, because this is a cue also for swarming. If there's a lack of E-beta-osmine suddenly in a hive, all the bees in the hive know that the queen is not functioning or she's run out of room to lay. Let me get to the spring turnover. And Al Avatable published this back in 1978, showing how in late winter, the population of adult bees begins to decline as the amount of brood goes up. You see this here in Loy's data right here, that these bees, winter bees, are almost immortal so long as they don't start brood rearing. But the moment they start brood rearing, they start dying very, very quickly. They got about 30 days and they're all going to be dead once they start brood rearing. And we get what's called the spring turnover, that they need to see their population, all this blue and purple just disappearing. That needs to be replaced by this red and orange of their replacement bees coming in right here. Yeah, you got a question? Yeah, unmute yourself.
Why are winter bees in danger once they start brood rearing?
They change, it's not danger, they change their physiology.
Oh, really? Okay.
Yeah, so they go from that diutinus, that long-lived physiology and no flying, to going into nurse bee physiology. And as soon as that first round of brood comes out, those young bees displace them and they shift to forager physiology and they die very quickly.
Ah, okay.
Yeah.
Thank you.
Yeah. And so this spring turnover is, especially out in the East Coast, we hear a lot of colonies collapsing. Colonies don't die from starvation during the winter. They die during this spring turnover right here. If they start rearing brood at this point, and then you get bad weather moving in here, if you don't intervene and give them supplemental food, these colonies can very quickly starve or run out of protein, which inhibits the ability of these long-lived winter bees to rear their replacements. If they can't rear their replacements, then the colony is toast, cannot recover. We see this out in the almond. This is almond orchard just beginning to come into bloom. And people bring their bees out of winter storage, bring them in here, and then suddenly all those winter bees start foraging and they just wear themselves out and these colonies collapse right in the almond bloom. Way better to bring them into almonds after they've already got plenty of sealed brood of emerging bees in the hives. So here's a bunch of old worker bees, nice rounded brood, but what you can see, there's barely enough worker bees to keep this amount of brood warm. That's often a trade-off at that point. Notice there's only a minimal amount of protein reserve here, a little bit of bee bread. They're all in. They're eating everything they get coming in during the day and putting it into their replacements. And then they must deal with pathogens. Now, your different pathogens, there's a difference between a pathogen and a disease. So all the viruses, the bacteria, they're potential pathogens, but they don't necessarily cause disease. So you can find those pathogens in the hive all year long, but that doesn't mean that they are causing disease. So typically, springtime, you'll see European foul brood and chalk brood. Nosema is a springtime issue when there's a pollen flow. So as soon as there's pollen, you're going to start seeing Nosema counts go up, but that doesn't necessarily hurt the hive. Then American foul brood, pretty much any time during the season, but usually during the middle of the season, you don't see disease issues. And then varroa and bee-forming virus become serious issues in the late summer, and then finally the paralytic viruses. And if you have a combination of paralytic viruses and nosema, that's very problematic for the hive. And this is a nosema. This is a picture of a bee gut, 400 times magnification. This is a tracheal tube, breathing tube. You can see the striations of the gut tissue right here, the epithelial tissue. And every one of these little white dots is a nosema spore. This is how strongly infected the gut tissue of the bees are. But amazingly, if they're getting plenty of pollen in their diet, it's not that big a deal for the bees. when it becomes a big deal, when the adult bees, the proportion of them start to get infested. If 10% or 20% of the adult bees are infected with nosema, not a big deal. You start getting up to 50, 60%, then they become a big deal. So here's a frame in February when we still get frosty nights. And you can see this was a 3 frames of bees, 3-frame colony. But look at that frame of brood, big, healthy frame of brood. But one glance of this colony, this was taken back during CCD. And you can say, oh, this colony is about on the verge of collapsing because this many adult bees could never have kept that much brood warm on a frosty night. So that tells you that attrition is exceeding recruitment in this hive. They're right at the spring turnover. And if they can't keep, these workers can't keep this brood warm on a frosty night, then the brood will die and that colony will die. And this is just what happens right here. You can see the ring of pollen that was around there, how big the brood nest was the day before, a couple of days before. On a frosty night, if you have a high proportion of bees infected with nosema, they die off and they're unable to maintain thermoregulation. And then these bees, then the next day, will clean out all the freeze-killed larvae and pupae, but this colony will not be able to recover. And this is typical signs of a nosema collapse right here, where you see that there was a big ring of brood, but they just could not make that turnover there. The other thing to understand about nosema is dysentery can transmit nosema spores, but it is not a sign of nosema. This is a sign of dysentery. That's an issue of water balance. So people are often confused thinking that dysentery indicates that the colony has a nosema problem. They're totally different problems, although dysentery will spread nosema spores. Did you have your hand move, Mike?
Yeah, are there any supplements you can feed the bees to assist or prevent nosema infections?
The only one that really works well is the fumagillin, Thymol to maybe a little bit. There's a couple of proprietary products, Nosavit, which has some good supportive data. Another one, Hive Alive. I need to see some more supportive data on that one. So those are possible ones. If you feed them pollen sub, nosema counts will probably go up because nosema thrives when the nurse bees have a bunch of protein in their guts. That's
right.
So, but it means that, well, let me go on there. So
okay.
I'll return to that question. Dysentery is just a sign of water balance, not of nosema. The only way you can tell if a colony has nosema is by putting them under the microscope. So here's 400 times magnification. Here's a typical bee hair. Here's pollen grains right here, and these are nosema spores. So this is a moderately, this is a squash diluted to 1 milliliter per bee. So this shows a moderately high infestation level right here. Here's the thing. Nowadays, Nosema serrani does not have much effect on a well-nourished colony. But if you have a large proportion of the bees in the hive infected, then it will slow down colony buildup and affect honey production. So if a colony is lagging in buildup, then suspect nosema. You could do a gut squash, look under the microscope. Otherwise, I wouldn't suspect nosema. Okay, once the colony gets past the spring turnover, it can go into the linear growth phase. Let me get some water here. And in this phase, when there's plenty of pollen, they can grow very, very quickly. There's some data from Nolan in 1925 showing the buildup of package bees. And you can see it's just a linear buildup, very, very, very rapidly. Again, until you reach about 40 times the queen's egg laying rate, and then the colonies stop building at that time. So they'll build up linearly for about 2 months, very rapidly. So it takes about 8 weeks for that. Once a brood nest is established for a colony to reach a full population. And you can show on Lloyd's data the same type of thing, this linear buildup right here. Now these colonies can grow very fast. Here's a colony that we're just putting a second honey box, empty box on from the buildup into. So this is a 10 frame, a split from the springtime, built up very, very strong, but they're living hand to mouth. Realize also there's a very rapid turnover of these bees. Roughly every month, you have a complete worker turnover. The average age of a bee, longevity of a worker bee is about 35 days. So that means you got to replace all your workers every month. So those people who say, oh yeah, my bees get to recognize me. Well, they're not going to recognize you if you're not in there at least once a month, because they're all going to be dead by your next visit. This requires 1 to 2 pounds of pollen every week coming in just to replace this worker turnover. So here's a colony living hand to mouth, growing just fine, but very little leftover pollen at the end of each day. By morning, this may all be gone also. And then if it rains, and we get lots of this out in my area, we get these springtime rains, late winter rains that just wreak hell on the colonies. They're building up, and then it rains, and they immediately start to go into protein dearth. And the nurse bees, within an hour of doing experiments, putting sprinklers on hives, they'll start cutting back on the amount of jelly that they feed to the larvae. You can see here with Lloyd's data, there was a hiccup right here in the brood rearing, the amount of brood. And I checked back in the weather data in 1976 for his location to see what happened. And sure enough, this was a cold rainstorm moved in Manitoba right here. And immediately the amount of brood rearing was reduced. That picture of the very populous hive I showed a couple of slides back, that same yard of bees, we had a freak snowstorm come in, and within 4 days, our colonies looked like this. They've consumed all their honey reserves, all their pollen reserves, eaten all the eggs up, and consumed all the young larvae. So the only thing they leave are the pupae ready to emerge, and these bees are right on the edge of starvation right here. And at this point of time, as my kids were growing up, I would treat my bees better than I treated my own children, because we'd say, okay, boys, put on your foul weather gear. We're going to go out and feed our beehives. But I've had people say, Randy, you're out feeding your hives pollen sub when it's raining and snowing? And I say, yeah, well, duh, because when it's warm and sunny, they take care of themselves. Your job as a beekeeper is when conditions are bad to get out there and take care of your livestock. So yes, we often spend we'll often go 30 days straight working bees in full rain gear here in California in the springtime. This is not unusual. This is normal beekeeping attire for us in the springtime when it's raining and freak snowstorms, out feeding our hives, because they need this to build up. And we'll also feed them sugar syrup to stimulate a nectar flow if they don't have enough reserves of honey in the hives. Okay, hive inspection. Generally, just go to the upper box of a double deep, pull out a center frame very carefully so you don't crush the queen. I take one look at this comb and I say, this hive looks just fine. We obviously queen right. We obviously have very high rate of larval survival. We have a band of bee bread here, so they're in good nutritional shape. They have a reserve of honey stores around, nothing else to learn. Slide the frame back in, close the hive up. The bees hardly even know you've been in the hive and you've done enough colony inspection. If I see other signs, a spotty brood pattern, lack of pollen, lack of honey, then I'll do a further inspection. But other than that, that's all we need to do. Okay, let's see what I got. So here's healthy brood, nice solid pattern here, meaning indicating good larval survival. Spotty brood, this could indicate disease. In the springtime, this would indicate European foul brood. Later in the season, it means that there's poor nutrition and the nurse bees are following the queen and eating the eggs up as fast as she lays them. Anytime you see the larvae with different aged larvae next to each other, that's a sign of there's something wrong. You've got some kind of disease affecting the larva. The larvae start to die, they get cannibalized by the nurse bees, and the queen comes back and lays another egg. So this could be poor nutrition, this could be disease, often European foul brood. There's chalk brood right here, very distinctive symptom of these hard mummified larvae with this oval of dark brown right here. Nothing to do about this if it persists, just requeen. This is a fungus that will go away, fungal disease. European foul brood, I'm seeing much more of this in recent years than we used to see. The larvae typically twist in the cells like this before they die, turn kind of yellowish, and it's only the younger larvae. There's no sunken cappings with European foul brood. Luckily, most strains of European foul brood still respond very well to a treatment with oxytetracycline. And we use it only spot treating. When we see it, we don't do any preventative treatment or prophylactic treating with antibiotics in our operation.
And do we know what causes it?
Yeah, Melissococcus pluton is a bacterium, and then there's secondary bacteria that move in along with the European foul brood.
Okay. Somebody asked what the green grass was for, but I'm not sure what he's talking about.
You know,
What
it's
it- can you
amazing to
please
me
explain?
how often I get asked that question.
Ah,
We use that as a marker. So if somebody's gone through those hives, hefted them, and said all the lightweight ones will need to mark for a bottle of syrup. So what kind of marker do you put on? You put some green grass on. Next time you come back to that yard, there's no more green grass.
right.
That means that marker was put on that day. So if you want a marker that's only going to stay on for an hour and then disappear, use green grass.
Okay.
Okay.
Cool.
So
Yeah,
we do that all
nice.
the time. Anytime we want to do, we typically have somebody going ahead in any bee yard, taking a look at the colonies, hefting them or opening them up, and then marking them. And the ones that have green grass on them, they need whatever we're doing. And the ones that don't need that. And there's no confusion next visit. It's not like a rock on the hive that you have to take off, because if it's not green, then it's an old marker.
I like it.
Generally, if you have mixed pollen colors, you have good nutrition in the hives. And here's a nice step down of larval age, going from older larvae to younger larvae, again, indicating good larval survival, and the amount of jelly around these larvae is indicating good nutrition. So what we do is we look at the amount of jelly around these second instar larvae. So here's a second instar larva right here. First instar larvae, the really tiny ones, don't have much, but you want to see the entire bottom of the cell covered with jelly, indicating good nutrition. So here's a larva. Larvae swimming in jelly. You're the best beekeeper in the world if you call it looks like this. These bees can handle almost anything, because you're going to have lots of very good recruitment of new, well-fed worker bees. Here's larvae only being fed just enough to keep them alive. So these bees will turn into normal looking workers, but they will have a different physiology and different behavior. They will mature and shift to foraging earlier than well-fed bees that were well-fed as larvae will do. Again, here's well-fed larvae, lots of jelly, and poorly fed larvae, very little jelly. For us, we found learning how to just assess colony nutrition by looking at the amount of jelly around the larvae to be incredibly important to us. Very, very helpful. So you don't have to ask what the nutritional status is of your hive. Just look at the amount of jelly. The nurses cut back on jelly when nutritional status is poor. Okay, so now we've gone past the spring turnover, past the linear growth phase. The swarming impulse happens when you start to max out on your amount of brood in the hive. So you have lots of sealed brood in the hive right here. And especially when you have backfilling of nectar, so that there's no more room for the queen to expand, and she has no room to lay any eggs. When she can't find a place to lay eggs, 4 days later, there's no E beta osmin, there's no young larval pheromone in the hive. And the bees go, oh man, it's time for us to make queen cells right away. That means either the queen is failing or we are running out of room. In either case, we need a new queen. And if we're running out of room, that means it's time for us to swarm. So when the colony feels that they've packed the cavity, filled the cavity, there's no more reason to exist other than reproduction. And they will keep swarming until there's room in that cavity for whatever queen is in there then to start laying eggs again. And one of the things you can do is you can do a manipulation of the brood nest, moving the drawn combs that are below the brood nest up above, reversing the brood chamber. This works very well for reducing swarming. Anytime you put drawn comb above. But if you start seeing on your second box, you start seeing all these queen cells, now it's too late for do anything. You need to make splits right there. And That's what we do. We specialize. We make about 3,000 nukes every spring. We set up a semi-line and just split our colonies and crank out nucleus hives, starting all of our hives fresh each year. If you don't do that, this is colony reproduction. You want to avoid swarming at all costs for a couple of reasons. Number one is you've just lost your honey production by the swarm. Number two, you now have a competitive colony out there trying to establish to compete against your hives. Number three, leaves most of the mites in the parent hive. So now the parent hive is just really mite-ridden. Not many of the mites leave with the swarm. And number four, if this colony does establish successfully, it's going to become a mite diffuser later on in the season, and you're going to have to deal with the mites drifting from this hive back to your hive. So avoid swarming as much as you possibly can. Now, at the time of swarming, you have frames full of sealed brood. When they're sealed brood, the pupae are stacked like cordwood, very tight. But when they emerge, notice a single adult bee covers about 3 cells. So that means there's a 3-to-1 expansion. So one solid frame of sealed brood will turn into 3 frames of workers when it emerges. And this is why a colony, after they swarm, can very quickly rebound their population from all this sealed brood in there. Then we get to the main honey flow. And ideally, you reach peak population right when the main honey flow starts. The other thing to know is the more populous the colony, the better a ratio they have of foragers and mid-age bees to nurse bees, and they are more efficient at producing honey. So larger colonies are better honey producers than one larger colony than 2 smaller colonies. So you really want to, what we do, we'll combine weaker colonies at the beginning of the honey flow to get greater honey production from these large colonies. Then the honey flow starts and you get nectar shake. You pick up a comb, shake it, and the nectar comes pouring out. At this point, at this energy interface, the bees are now, as soon as any of these pollen bee bread cells get consumed by the nurse bees, then the mid-age bees pack these full of nectar and they force the brood necks back downward. And they'll fill these combs in here also temporarily with nectar. When they run out of combs to store the nectar, that tells them they need more room. So they'll start producing white wax to build more comb. And this is the beekeeper's signal to add foundation or extra space. Yeah, Mike.
When you're making your super hives by combining for honey production, do you ever go larger than 2 deeps?
Some people do. We don't, but some people do.
Okay.
If you're in a really intense, like up in Canada or the northeast, where you have these, you can make a 300-pound honey crop. Yeah, then you can make these monster colonies.
Okay. And do you use excluders at all?
That would be up to. I'll tell you. One rule for beekeepers is beekeepers have this innate drive to take the simplest aspect of beekeeping and making it as complicated as possible. This is across the board. Beekeepers will I can tell them the simplest way to keep bees, and they will make it harder and more time-consuming, more expensive at every single chance. So when you say, do you use, there are people who will, I've read the 2-queen systems that are so complicated, it makes my head spin. So it depends on what your joy is. If your joy is to make it as complicated as possible, man, use everything possible. Okay?
Okay.
So this is a sign that it's time to build a new comb. They've run out of space in the hive. And this is when you can draw foundation. There's no tricks to drawing foundation. You can only draw foundation when the bees are making white wax. Otherwise, you're wasting your time. One thing to do, though, is to make sure there's not a honey band above the brood nest if you're putting foundation over the top, because the bees are loath to cross this honey band, especially through a queen excluder, and draw foundation. You want to have brood right up to the top bars where you're going to be drawing foundation, so you can reverse your brood chambers. And then the bees maintain a balance between the population of mid-age bees, which are your nectar processors and comb builders, and your foragers. And I'm not going to go into that, but there's feedback loops. At that point, brood rearing is deprioritized. They may fill all the brood cells with nectar. And look at what happened to Lloyd's up in Manitoba when the honey flow started. Bing, queen was just shut down. The bees don't need any brood rearing during the main honey flow because any bees, it's going to take 3 weeks before they emerge and another 3 weeks before they become foragers. So by that time, the honey flow is going to be over. There's going to be extra mouths to feed. So the colony will deprioritize brood rearing when the main honey flow starts. But you can trick the colony by adding extra space so they feel they don't need to swarm. They think they get the feeling, oh my God, there's all this empty comb. We better put on a big honey crop. Here's Tim Ives in Indiana, where he's getting 350-pound honey crops, surrounded entirely. As soon as the other side of these trees, it's nothing but neonicotinoid treated corn and soybeans as far as the eye can see in all 3 directions. And he puts on these huge honey crops. Okay, then after the main honey flow, your late summer decline, nothing to eat, and the colony is going into survival mode during the summer pollen dearth. Very little recruitment, so the colony population starts to drop down, and attrition exceeds recruitment. And you see in Lloyd's also in Canada, they don't want any more bees after this point because they're just going to, they're non-productive. There's going to be extra mouths to feed. So they want this, the colony wants this population of bees to go down. And you see typically this would be late summer, some fresh honey right here, but look at the poor brood pattern. No bee red at all right there. So protein deficit, the nurses will follow behind the queen and they'll consume the eggs again as fast as she will eat them. And you get these spotty brood patterns, not disease, just poor nutrition. If you wish, if you have a reason to do so, and you may not have a reason to do so, you can rejuvenate that colony by getting more recruitment going by feeding them pollen subs. So those of us who are getting ready for almond pollination, we start in September initiating brood rearing by feeding them protein. If you're not interested in going to early pollination, you wouldn't want to do this because that just means your colony is going to swarm early in the springtime. So again, look at your goal and work backwards from your goal. Then varroa. You can, there's a free model on the internet, Randy's varroa model. You can punch in your type of colony, and it'll show you your mite wash counts and your populations of mites, of bees in the hive, of sealed brood, and the percentage of brood infested by varroa mites. You want to avoid this red crash right here. What happens is, notice the varroa mites never really explode. What explodes is because the amount of bees is going down in the late summer and fall, and whereas the mites keep going up, your infestation rate suddenly explodes right here. And this is when deforming virus becomes a problem in the hives. And you start with 100 mites here, it's easy to build up to 10,000 mites in the hive. And when it starts to, mites start to build up, you're going to see things like varroa sensitive hygiene, where they start chewing out the infested pupae. Bald brood, and we're actually selectively breeding for this. This is a varroa fighting trait that resistant bees use to fight varroa mite. You may start seeing bees emerging with deformed wings, and you'll start to see larvae and pupae dying from virus in the cells. And that's because this is your mites per 100 bees right here, this red plot going up. And this is your prevalence of deforming virus or the paralytic viruses. They track the infestation rate of the mites. So you want to keep your mite infestation rate way down here to avoid this virus buildup. And once the percentage of infested pupae passes about 25–30% in a hive, the colony is toast, it's walking dead. And you start seeing this happen. We're approaching parasitic mite syndrome here. Bees with deformed wings, bees unable to emerge, larvae and pupae all dying. Looks pretty ugly in the hive. And this is the #1 cause of colony losses these days. It's lack of control of varroa mites. And you can do a post-mortem. If you see these white fecal deposits at the tops of the cells where the last of brood was, that tells you you had too many varroa mites in the hive. There's another picture of those white fecal deposits. And it's not only you, when you're calling it high mite levels, look at the mites on the back of the bee and mite on the side of the bee there. These bees are drifting all the time from hive to hive. They drift easily up to half a mile away to other hives, and they share the mites with all the other hives in the neighborhood. As far as varroa control, the number one technique used by beginning beekeepers is called wishful thinking. And we have plenty of data showing that wishful thinking has never done anything for varroa mite management. Move on to something else that actually works. Here's somebody who's done mite control, 3 low efficacy treatments throughout the season and just kept their mite level down low. Most areas you can require 2 to 4 effective treatments per year. You want to get down close to 0 for good wintering. Here's what we do in our operation in California, and this is local for us. Our colonies go to almonds, come back very big. We split them. We create an induced brood break. We hit them with an oxalic acid dribble. They have low mites then all the way through the honey flow. Towards the end of the honey flow, we'll give them a knockback with a single formic strip right here to buy us time while we pull the honey right here. Then we'll give them a time all treatment and then start feeding pollen sub early September and the oxalic acid dribble when they go broodless. And we've repeated this year after year and it works just fine. No synthetic miticides, no residues in the combs. It works quite well. The long-term solution is for us all, eventually we'll be having mite-resistant bees, and we are very much involved in a breeding program for this. And these are, we're using the phrase now, zeros are heroes. These there. Here's the mite count with no treatment of this hive in July, August, September, November. There are colonies in our operation that just are immune to varroa mites. So the only, every year, we only breed all of our queens off of colonies that are controlling the mites by themselves. It's really tough to lock in heritability. We've been doing this for a few years. It's getting better, but we still cannot say that our bees are mite resistant. Then the autumn pollen flows start, and the bees often store bee bread beneath the brood nest, so they have these reserves of pollen if you're in an area where you do get autumn pollen flows. And it's these bees right here that are reared at this time that are going to become these winter bees. The summer bees are all going to die off. And some interesting research from Heather Maddle is showing that only in the bees that emerge late in the season, in September and October, instead of dying very quickly, here's the survivorship curve. If they emerge late in the season, they have this longevity until they start rearing brood in March, and bingo, then they start dying again very quick. So you want to be really make sure the colony has good nutritional status at this time, so these long-lived winter bees can make it through the winter. So first we control mites, and then we stimulate brood rearing and give them supplemental protein shortly before them going into the winter cluster. If the colony does not consume the pollen sub at that time, we just walk away from it. Not worthwhile even messing with. They're not going to make it through the winter, so don't waste time on them. Then you get your first frost, and no more pollen coming in, and they knock off brood rearing. And then you have this fall turnover where roughly about half the bees in the colony, the older bees, just fly off to die. This altruistic self-removal from the colony. And colony strength drops about half suddenly, typically in November. These emerging bees, there's no larval pheromone, there's no brood rearing going on. So they, instead of becoming nurse bees, they become the long-lived diutinous bees, build up their fat bodies in their bodies and hunker down to survive until pollen starts coming in again. And then here's this fall turnover with this about 50% of the workers just disappear. Then you form the winter cluster. If you quick open it up, it looks like a solid ball of bees, but if you give them a puff of smoke, what you'll see is it's a tight shell of bees and then tropical environment in the inside where the bees are free to move. And it's an airtight cluster here that they actually the amount of CO2 goes up, the amount of oxygen goes down, and they control this, the humidity and the temperature in the middle of this core, exhausting most of the moisture out of the bottom of the cluster. So there's big questions about whether top ventilation is helpful to hives or not. Top insulation is very helpful. You can see the heat loss of these hives here. So 1.5 inches of styrofoam above or below the cover will help these colonies a lot. And then we get into these long-lived winter bees here, very low attrition. I mean, look at the survivorship curve of these bees. It's just about flat here in Manitoba from November until March. Very little attrition of the bees. And at this point, what we have is no protein reserves. This last of these larvae are going to be emerging as winter bees. Now, you may get midwinter initiation. Most brood rearing, most colonies will rear some brood during the winter, but it seems to be mainly an issue of water balance rather than a need to rear more bees. And this needs to be studied more. Then here out in California, we'll stimulate late winter brood rearing in January to build our colonies up for almonds. A friend of mine, Keith Jarrett, builds these huge colonies by feeding all winter long, and he shakes bees for sale in the springtime and sells them to other beekeepers, sells bulk bees in January, all on pollen sub. Then the first tree pollens come back again, and we start the cycle again. And that's it.
So what's your preferred way of feeding for the winter? I may have missed it.
Preferred way for feeding pollen sub?
No. For carbohydrates.
Well, first place, you don't need to feed at all if you don't take the honey.
Right.
You leave enough, and so we don't do much fall feeding. We just leave them their own honey on the hive. Now,
Okay.
in many areas of the world, beekeepers harvest every drop of honey and then feed back sugar syrup. It has to be done when it's warm enough for the bees to process that syrup and convert it into sugar honey. So that's up to the beekeeper. And there's lots of different preferences. All have read an article years ago. It's on my website, the pros and cons of different ways. So I don't make any recommendations, Mike, at all. I'm happy to give hard data, show you the results of trials, but every beekeeper, they can use that to make their own decisions, management decisions.
Fair enough. So how are things going with the OA shop towel method?
Funny, I was just speaking with a large commercial beekeeper today who called up, who's been experimenting with it and going well. Tomorrow we're hoping to do a bunch of alcohol or detergent washes, a couple hundred hives, and collect final data on a large trial I'm running where we put different numbers of oxalic acid and glycerin strips on 2 different matrices into hives based upon their starting mite counts, looking to figure out what's the optimal amount to apply to a hive to get good mite control. Preliminary results are indicating that we want to be putting about 3/4 of a full Swedish sponge in, the equivalent surface area of that. But in a couple of days, I'll have a whole lot more hard data on that. Looking very, very promising. Still not any registered products, and I'm still not sure what USDA and EPA are doing as far as trying to, what my ideal would be to have EPA added as an approved treatment method so that beekeepers could make their own oxalic acid and glycerin strips. I'm not sure the EPA is going to do that.
Okay. And what's your take on the most recent information that it's safe to use OA with Superzon?
Yeah, well, some of that's just based upon data that we got, where we actually took honey samples before and after treatment.
Okay.
And there's plenty of research out of other countries where they've done the same thing, taken honey samples before and after. Oxalic acid, FDA and EPA are not at all worried about human exposure to that. If you eat a serving of spinach, you can get up to 1 gram of oxalic acid in a single serving of spinach. The micrograms are milligrams that you might get in a serving of honey are nothing compared to what you get in your normal natural diet if you eat any vegetables.
Okay, great. And excuse me, is there a reason that you prefer the dribble method over vaporization?
Yeah, what's interesting is we've done dribble with our operation for years. And then a couple of years ago, I got some vaporizers, and we ran some trials with the vaporizers, and the crew used them. And when we were done, every member of the crew said, "Why in the world would anybody use a vaporizer? It costs more, it takes longer, and it's way more dangerous to the operator."
Yeah.
So I can see if you're in Canada where winter comes on suddenly, and you don't want to crack the propolis seal in the hives, you can just shoot it in the entrance. Other than that, 2 of us routinely go out and dribble 400 hives a day for 2 of us in multiple yards, going from yard to yard. And one day we can easily dribble 400 colonies. It's totally safe. We don't have to wear any protective gear. We don't have to worry about inhaling it. It doesn't cost much. There's nothing big to drag around. Very cheap and equally efficacious. So that's up, again, that's up to the beekeeper. Like I said,
Yeah.
beekeepers have this tendency to make things as expensive and difficult as possible.
Right. And one of our other guest speakers said that beekeeping is 10% facts, 90% opinions.
Yes.
So, is there any temperature below which you wouldn't do the dribble method?
You want the bees ideally in cluster, but not in a super tight cluster. So if
Right.
they're in a tight cluster, a number of beekeepers, including ourselves, you come into the yard, you kick each hive first, and then you go back to the beginning, and that breaks the cluster. So the bees are moving around a little bit, and we open them up and dribble them. So typically we're doing it above freezing temperatures, but we want to be down there, at 40, 50 degree range works really well.
Okay. Okay, good to know. Yeah, so your climate is kind of similar to ours then, at least for some period of time.
And typically that's when the bees are going into winter cluster in most any climates. Until you get way far north where winter can come on very, very suddenly, you got plenty of time in the fall as the bees are getting ready, are curtailing brood rearing to before it gets too cold to go ahead and give them a dribble.
Okay. And would you only use it when you think you're broodless?
It's not nowhere near as efficacious when there's brood in the colony. So when there's brood in the colony, you need to do multiple treatments or use the extended release. I just got a data set this morning from a beekeeper who treated with oxalic acid vaporization every day, nonstop for about 25 days.
Oh
and
my God.
I'm looking at that, and he's still dropping mites after 25 days of a day after day treatment. So I just wrote to him this morning. I haven't read his response yet of whether he was using 1 gram or 2 grams on the vaporization. But that kind of data is making me question those people who are claiming that vaporization is efficacious when there's brood in the hive.
Wow. Okay. Because, excuse me, 2/3 or more of the varroa are under the cappings, right?
That depends on your time of year. If you look at my model, my mite model, it'll tell you roughly what proportion are under. So at swarming season, you got maybe 80% of the mites are in the brood. The rest of the year, it's usually 50 to 60%.
Okay. All right. I think you've converted me to dribble, though. All right. Anybody?
Yeah, I'm wondering, did we get any people coming in from the
I didn't see an uptick. In fact, it's actually dropped off a little bit. Not not right at 8:30, but I didn't see any additional people show up.
Okay.
So, everybody, any questions? This could be your last opportunity for a year. If we can get Randy to come back. All right. Well, that was great. I was a little concerned from the title that it might be too basic, but my worries have been allayed.
Good. It's amazing how often I hear that response from experienced beekeepers.
Yeah. Yeah, it's, sometimes I'll see the same presentations multiple times, and all it takes is like one off script question and
Yep.
all these doors open and these new pieces of information
Yeah.
emerge.
One of the things that some people find annoying about me is I say any beekeeping question that can be answered with a single sentence, they're only giving you a fraction of the answer. How deep do you want to go? So
Yeah.
I'll do classes and I'll show up and say, it's just gonna be question and answer. No format at all. What's the first question? And we started some classes at 9 o'clock in the morning. And at 11:45, I said, well, I'm still answering the first question. We're gonna have a second question after lunchtime.
Nice.
If somebody is knowledgeable, many of the very simple questions which get one answer, if you want a simple, straightforward answer to any beekeeping question, always ask a second-year beekeeper. They will give you the most definitive, simple answer at all.
Right.
If you ask me a simple question like that, you may get easily a half hour to an hour answer.
That's great. Okay, so we're getting a lot of compliments, but no questions. Oh, wait. Okay, here's one I missed. Hop guard, 2 to 14 days. Okay.
Okay, so very interesting you should ask that. I did a trial of hop guard last year when there was brood in the hives, and it failed utterly. But I didn't want to embarrass the manufacturer, and I know the manufacturer's rep, who's one of the are the biologist on the company. So I wrote to her, and the label was unclear. And I'm not the only researcher who misread the labels that you needed to repeat the treatment during the summer if you apply it. So I said, okay, I'll run another trial this spring. And I'm more than about 2/3 of the way through that. We just took, I applied strips 2 days ago, and I'll take final mite wash counts. So what I'm doing is I'm doing it with a, they didn't really have a good recommendation. So what I'm doing, the trial I'm doing is I have a control group of hives, untreated. One group of hives, I'm applying Hop Guard 3 every 7 days. The other group, I'm applying it every 14 days. So 3 repeat treatments at either 7 days or 14 days. I took starting mite counts at 21-day mite counts, and I'll be taking 42-day mite counts next week. And then I'll have some good data on colonies with brood. What's the most efficacious treatment with Hop Guard?
When you do these tests, excuse me, do you look for colonies with particularly high infestation rates or a particular infestation rate?
That's a really good question. If you want to see really good efficacy and you're using a mite wash to determine efficacy, what I found is across the board, the higher the initial mite count, the better the efficacy of most treatments. It's much more dramatic. You get a very high efficacy. If you start off with low mite colonies, it's really hard to get good efficacy. So when I'm doing these, if they're going to be like a 42-day trial like this, if you start with too high a mite count, you can have colonies collapsing before the end of the trial. If you start with too low, you don't get very good efficacy. So in this trial, I looked for colonies that had moderate mite counts. Most of them like in the range of for a mite wash in that 7 to 20 or 25 range, so that they would make it to the end of a 42-day trial. And then I took the ones that were mid-range and used those for the control group. then I ran the treatment on the ones that were on the lower range and the higher range to see. Then you get to see both efficacies for low mite levels and the high mite levels. So it's the way experimental design is everything, and that comes with practice of doing these field trials and seeing, and then looking at your data very closely afterwards and seeing how easy it is to get data that is influenced by how you set up the experimental design. It's been a really big learning curve for me. So I try to share that in my articles of what I'm learning.
Mm-hmm. Okay. Somebody has a question. Mary asks, some of my hives are terrible at drawing comb. Can you address again the reason why a hive during the honey flow will not draw new comb but fill in the brood area?
If the new comb has a honey band between where the brood area and where you placed it, they're going to fill in the brood area first generally. They don't like to cross that honey band. So one thing possibly to do is to reverse your brood chambers or to move a couple of frames of brood that do not have honey band above them to the upper box right immediately below the drawn comb that you're putting in there.
And here's one on mite wash. Excuse me. Do you ever examine the washed bees and see mites clinging to the bees after the wash?
Yeah, what we do is we validate any method that we do. We'll do 100 mite washes and we'll save the bees in a bucket, and then we'll rewash those bees very, very vigorously and see if we get any more mite drop from them. And we want to see essentially zero. That way, we're always looking for at least 95% recovery.
Okay, here's one of my own. Is there any value to washing bees after a colony collapses, or is there anything to be learned from that?
Well, there's no bees once the colonies collapse.
Well, okay, you're dead bees. Dead bees.
No.
No.
Yeah, I don't know. Did you learn anything? Because the mites are not going to want to stay on a dead bee.
Right. Okay. Well, I have done that, and I wasn't expecting anything decisive, but I have had colonies where I'd get about 20 mites. So that tells me that they must have been like at 60 or something before they died.
Sure.
Right. Okay.
Yeah.
All right. So, okay, it looks like that's about it. And we're about right on time also. Well, thanks very much. I hope you'll be willing to come back in a year or less and do something else, do the next topic up. All right. I appreciate it. Thanks, Randy.
Okay. Take care, everybody. I'm happy beekeeping to you.
Yep. All right. See everybody next month, hopefully in person.
All right. Wash your hands.
Thank you.
All right.
Bye.
Bye now.